Thanks, folks, for coming. We're honored to have Dr. Rob Knight join us today. He's a well-known bioinformatics researcher who has collaborated with both Professor Rosing and myself quite a bit over the past few years. And he does a lot of really groundbreaking work in the world of microbiome research. So yeah, thank you for joining us, and please take it away.
Sure, absolutely. Thanks for the invitation to do it, Nima. And it's been a great pleasure working with you and Diana over the years, and lots more cool stuff that we can do together as I hope the students appreciate from this. So what I'm going to talk about is the coming AI revolution in diet probiotics and the microbiome, and how the field has progressed from simple techniques that were applied mainly in the lab to things that we can increasingly use the kinds of algorithms that Nima is working on and the kinds of hardware approaches that Diana is working on to really make dramatic progress to make it useful at the individual level. The work I do is actually useful, so I have a bunch of interactions with companies that I'm required to disclose. I also made a bunch of the illustrations in this talk with AI. So this one, however, was not made by AI. This was painted by the Italian artist Arcimboldo back in 1591. And this is one of the first representations of this idea that we are what we eat in art. The pithy statement encapsulating that, though, wasn't formulated until the philosopher Feuerbach in 1863 formulated it in German as Der Mensch ist, was er ist, which is a great pun in German, less so in English. It just says the person is what he eats. And shortly after moving to San Diego a decade ago, I had the pleasure of meeting Jeff Bland at one of his events from the Personalized Medicine Lifestyle Institute. And I was particularly struck by this quote from his talk where he said, "Food is a language that speaks to our genes." So think about that for a moment. What he means is that our gene expression is fixed at birth, but fixed at conception rather. But what those genes do depends tremendously on what we eat in terms of how they're expressed, even the particular RNAs and proteins that they make. And in terms of that gene expression, you probably all heard this idea that you should eat the rainbow. And we're increasingly getting a sense that this language of food is not only a language of color, but also a language of chemistry. So everything from the carotenoids that make the carrots orange to the anthocyanins that make the berries purple. These are specific molecules that we're increasingly finding have an influence on the activity of gene expression, both from our own genomes and from the genomes of all the microbes that inhabit us. However, when I take my kid to the source of food closest to our house in Pacific Beach, regrettably, what that source is this. And you can see a lot of bright colors and strong flavors there on the shelves. And ironically, their motto is too much good stuff. But the suspicion I have is that they've systematically stripped out all of the good stuff from food processing and replaced it with synthetic analogs that have completely different chemical properties and impacts on our gene expression than that rainbow of colors in the brightly colored food I showed you. And there's more and more concern about ultra-processed foods in particular. So this article from the American Medical Association a couple of years ago is entitled "What Doctors Wish Patients Knew About Ultra-Processed Foods." And it's absolutely full of things that you don't want to hear doctors saying, like the health consequences of ultra-processed foods are dire, and that those concerns include type 2 diabetes, cardiovascular disease, and dementia, all of which you definitely don't want, right? But fortunately, if you're reading articles in the popular press, there's a very simple solution to all of this. And this article that came out at about the same time has a great headline, right? "Cheese protects you from all causes of death, says science." And why would science say it if it weren't true, right? It turns out that humanity's long battle with the Grim Reaper is finally over. All we had to do was fight him off with the right piece of cheese. So I'm being a bit sarcastic about that, obviously, but the fact is that research in nutrition is really difficult due to the complex and highly multivariate nature of the data sets that you have to grapple with. And this paper from Darish Mazafarian and his colleagues in the New England Journal of Medicine, one of the most prestigious medical journals, that came out back in 2011 is still one of the largest studies of its kind, where they looked at 120,000 people over the course of 20 years to look at the impact of each food on weight gain and weight loss. And I'll be honest, the findings of this were extremely unsurprising, and I bet comply exactly with your stereotypes of what fat people eat and what thin people eat. So if we look at the food most associated with weight loss, that food is yogurt. And then correspondingly at the other end of the scale, so to speak, the food most associated with weight gain was potato chips. No surprises there. But what is perhaps surprising is the magnitude of the effect. So each serving of yogurt per day leads to a weight loss of only 372 grams over each 4-year interval. And the weight gain that you get from potato chips over each 4-year period, and this is for a serving every single day, is similarly modest. And so what this means is that if every single day you were going to eat a serving of chips, the worst food, and you gave it up and you ate a serving of yogurt instead, the best food from a weight perspective, you'd have to make that right choice 1,460 times in a row, and you'd only change your weight by 1.14 kilos, so just by a couple of pounds, which is not very impressive, right? Maybe you'll say, well, I'll just eat the chips then. But what this is masking is individual variation. So some people are going to make that substitution and lose 20 pounds. Other people are going to make the substitution and gain 18 pounds. And it all averages out when you look at the whole population scale. And this is true not just for weight, but remarkably for mood. So the Food and Mood Centre at Deakin University in Australia ran this amazing trial called the SMILES trial, where they got people newly diagnosed with major depressive disorder and put them on a Mediterranean diet. And they found that 32% of the people had improved depression symptoms, which is comparable to what you would get from drugs like SSRIs, so Prozac and so forth, and comparable to what you get from standard of care group therapy. And as a control to check that it was the Mediterranean diet specifically, not contact with the people doing the counseling, they did the same sort of thing on a low-fat diet, and that low-fat diet only had an 8% recovery rate. So that showed that it was specific to the Mediterranean diet. They did a follow-up to this called Healthy Med, where they compared the Mediterranean diet to social group therapy, which is standard of care for major depressive disorder, and found similar results, where the Mediterranean diet was highly effective. And there's an increasing literature on this. So this was a recent meta-analysis in the systematic review of whole dietary treatment in patients with depression. But I have to say that if you're studying this field and you read this paper, you yourself might get a little depressed, because it turns out that they had to read 1,863 studies to find only 5 studies that were useful for drawing conclusions from. All the rest of them were low quality, with flawed study designs or too small a sample size and so on. This points to a real problem with this field, where companies that want to sell you a particular food do an underpowered study on just that food, and then the results are not very useful. And the other thing I want to highlight is that although 30% of people recover on the Mediterranean diet, that still means that 70% of them fail. And so finding out who's going to benefit from a particular therapy, whether it's diet or drugs or cognitive therapy or what, and telling them early rather than letting them go through months of failure, is still a really important question. And so looking at the human genome for the key to this individual variability in diet and health has been a huge project, and literally it's cost billions of dollars. There's this whole field called nutrigenomics, which has had very modest results. And even if I have your complete 3 billion nucleotide genome sequence, I can't tell from your genome sequence what you should eat specifically. And so then the question is, are we even looking in the right place? Or are we like the proverbial drunk man looking for his keys under the streetlight, not because that's where he dropped them, but because that's where the light is shining on the human genome? And so then the question is, are the keys actually somewhere else entirely that we're not looking at? And so where I think that is, perhaps not surprisingly, is in the microbiome. So each of us has about 20,000 human genes fixed at conception and inherited from our mothers and our fathers. But the size of our microbiome gene catalog is somewhere between 2 and 20 million microbial genes. And so what this means is if we focus only on the human genome, we're ignoring 99% of our genes. But more importantly, the ones we ignore are the ones we can change. And it's important that we study them now because we're losing a lot of them. So when we work with people like the Hadza hunter-gatherers in Tanzania, what we find is a tremendous amount of seasonal cycling where they eat different foods at different times of year. So when I was visiting them, they were eating mostly honey and baobab fruit. I've never heard anyone say, I'm going on a paleo diet, so what I'm going to do is eat mostly honey and fruit for the next month. But that's exactly the sort of thing you would do if you were actually living a hunter-gatherer lifestyle. But what's fascinating is that when we compare them to populations around the world, what we find is that the microbiomes of each population automatically sort out from least industrialized on the left to most industrialized on the right. And this is coupled with a tremendous loss of microbial diversity. So entire major groups of bacteria that you see on the left-hand side, and all of these people living non-industrialized lifestyles disappear, and you see them replaced by a much smaller microbial community that comes in if you're living an industrialized lifestyle. And so it's almost like we have this rich inner rainforest of our gut filled with life and diversity, and we bulldozed it so that we've got a cityscape where just the pigeons and the rats survive. And I'm sure you've been taught about Rachel Carson's work from the 1960s that led to banning DDT, and her book Silent Spring that documented how DDT and other pesticides wiped out all kinds of life from throughout the ecosystem, including the birds, which were definitely not targets of those campaigns aimed at containing arthropod pests for the most part. My good friend and colleague, Marty Blazer, wrote this wonderful book, Missing Microbes. So you can see a signed copy on my shelf over there. He documents a similar loss of diversity for our inner ecosystems, where all kinds of things ranging from antibiotics to increased use of C-sections to low-fiber diets are depleting our microbial inner ecosystems. And one paragraph that Marty likes to show is this pair from the New England Journal of Medicine back in 2002, so over 20 years ago, documenting how over the 20th century, one infectious disease caused by a single organism pathogen after another, everything from measles to TB, declined. But at the same time, you see an explosion of all these different so-called non-communicable diseases, everything from inflammatory bowel disease to multiple sclerosis. And so the question is that depletion of our ecosystems contributing to that rise in these so-called non-communicable diseases that are changing much too fast for it to be human genetics changing, and which continue to rise into the present? So this might lead you to wonder how we find out about these microbial ecosystems. And I'm going to talk about three phases of how microbiome research has happened. The first phase is integrating evolution and community ecology, which involves some computational aspects. The second is going to be incorporating machine learning, which is far more computational. And then the third and most recent that we're just starting on now, and that you could contribute to if you decide to study this in college or in grad school, is incorporating AI, which is transforming our capabilities once again. So you might be wondering, what do I mean by community ecology? And all of this stuff came from the study of landscapes and was pioneered by Whitaker in the 1940s for the most part. And basically, the idea is very simple. The concept of alpha diversity, which is how many kinds of life do you have if you draw one circle around an area and count up, say, the number of tree species in it. You have beta diversity, which is a comparison between two different patches, and you're asking, are the kinds of organisms there similar or different? And then you have a concept called gamma diversity, which is all the diversity over the whole landscape. And this hasn't been applied as much in microbiome research as alpha and beta diversity, but it's getting more and more interesting as we start to study the microbiome of the whole body. So these concepts dominated microbiome research for a decade. And the data has evolved from just a few bits of information about each sample. So what you're seeing here is the result of taking a particular marker gene and running it on a gel where it will separate out by size or other properties of the sequence. And so you basically get bands on the gel with different intensities, and you can count the number of organisms that you could see with a relatively crude limit of detection. But that's all the information you get, and you don't know the sequences, and you don't know what they correspond to. And there's this alphabet soup of different techniques that I won't tell you, because all of this is obsolete, and you'll never see it even once in a lab at UCSD. So what transformed this was technology that we introduced in 2008 that let us take hundreds of samples and then sequence them together on a single sequencing run of what was then called next-gen sequencing. And so in this first study, we took samples from the human lung, from rivers across North America, from air samples, and from the most complex microbial community we know of on Earth, the Guerrero Negro hypersaline mat. And we sequenced them all together on this one run with this barcoding technology where we took Hamming error correcting codes and we encoded them in DNA, and we used that to be able to deconvolute what sample each sequence came from by reading out the DNA with the code and then the DNA with the sequence. And you can see this beautiful clustering by sample type showing that the technique works, but that's not what I'm going to talk about. I'm going to talk about the cost because that's what really revolutionized things. So in this study, we got 500,000 sequences, which doubled the total number of this kind of sequence for marker genes for bacteria that were available to the scientific community. And at the time, each sequence cost $8 with traditional sequencing. So if we'd done this the old way, it would have cost $4 million, so 500,000 times $8, and it would have taken a decade to do. Whereas with the then new 454 technology, we did it overnight and it cost $12,000. So that's pretty cool, right? And that's what enabled large-scale projects like the Human Microbiome Project, the Earth Microbiome Project, the American Gut Project, and so forth. But what's most amazing is that sequencing that cost $4 million in 2007 and that we reduced to $12,000 in 2008, today, if you do it at the facility at UCSD, it costs you just 75 cents, right? And so if it's going to cost you $4 million to characterize a sample, you're not going to do very many of them. But if it's less than $1, then hey, why not sequence your dog? Why not sequence the dust off your TV? There's all kinds of things that you can do. And this is what's leading to this flourishing of environmental microbiology in being able to just find out who's where in all kinds of different environments, ranging from huts way out in the rainforest all the way to the International Space Station, which we're writing a paper about right at the moment with one of the astronauts as a co-author. So to reduce that data, we introduced a computational pipeline called QIME, Quantitative Insights into Microbial Ecology, where the development for that was led by Greg Caporeso, who's now a full professor at Northern Arizona University. And when we apply it to the human body, this is a principal coordinates map of what are called unifrac distances, a beta diversity measure that takes phylogeny into account. You can see that it's like different continents with the mouth, the skin, the vagina, and the fecal material all being like different continents on this map of how the microbiomes are arranged. So each dot on this is all the diversity of a microbiome compressed down to just one point. And then 2 points are close together if they're more similar in their microbiomes, and then they're further apart if they're more dissimilar in their microbiomes. So from this sort of analysis using community ecology, you can tell the statistically significant differences between different groups. And we now know that the gut microbiome records a lot of things about you, like how you're born, how you're fed, what diseases you have, and how you try to treat those diseases, even things like how much exercise you do and where you live. And but we're talking about the gut microbiome. It's important to recognize that the gut is not Vegas, and that what happens in the gut does not stay in the gut. And that gut microbiome has huge impacts all through the rest of your body. So we now know that the gut microbiome affects digestion, as you'd expect, but also the immune system function, protection from pathogens, and even brain development depends on your gut microbiome. But it also has negative effects, including metabolic syndrome, including diabetes, liver disease, and so on, GI disorders, cancer, and various neurological and neurodevelopmental disorders, ranging from autism to Alzheimer's. So this interaction between ecologists and physicians has been very useful, but it's also been very frustrating for physicians who often have questions like, what do all these dots mean on these kinds of computer-generated dimensionality reduction plots? What do all these different kinds of diversity actually mean? Like there's a whole lot of different ways of measuring alpha and beta diversity that often give inconsistent results. And which organisms are really making the difference, which you cannot do with that kind of marker gene analysis. And then they also want to know, is this a clinical test? And everything I've shown you is, in general, research methods. You cannot get CLIA approved or FDA approved to do it as a real diagnostic, although there are people starting to work in that area. So we needed to do something more to make this stuff useful for physicians, to not just be able to tell that there was some kind of microbiome difference between groups, but to take it down to the individual level and ask, for an individual, what group am I in? So the solution to that was the second phase of microbiome research, integrating machine learning. And the key, and so we can now build predictive models for a lot of different traits related to health from the microbiome. And today, for example, I can tell you if you're lean or obese with over 90% accuracy based solely on the DNA of the microbes in your gut. So on the one hand, this is an awesome trick from the technical perspective, but on the other hand, we don't think it has a lot of clinical or commercial significance as a test for obesity, because I bet you can tell which of these people is obese without sequencing anything at all, right? But if you try to do that same classification task, lean or obese, from the human genome, you can only do it with 57% accuracy, whereas we can do it with 90% accuracy from their microbial genes, as Dan Knights, who was a grad student in my lab in Boulder and is now a full professor at the University of Minnesota, demonstrated as part of his PhD thesis back in 2011. And this shows you the noisy underlying data on the left. So these incredibly, so we're looking at the first and second time point, they're colored at this very broad level, the phylum level. And you can see that for the same person, the profiles at the first and second time point aren't even correlated. But what that leads to is a random forest classifier that over a broad range of sequence similarity cutoffs for grouping the sequences together into the same kind gives you 90% classifier accuracy. And this was taken a step further by Aran Segal and his colleagues at the Weizmann Institute, where they wanted to predict not are you obese right now, but what is your response to food? And so they got 800 people, hooked them up to continuous glucose monitors, fed them a defined sequence of diets so they could isolate the impact of each dietary item on their postprandial glycemic response. So basically how much sugar goes into their blood after they eat. And then when they averaged the values for all those people, they basically perfectly recaptured the tables that you could look up about the glycemic index in a book. But what they found was that the individual glycemic index, so how bad it was for your blood sugar individually for the same food, was all over the map. One of the stunning results from the study was most people think of rice as a pretty healthy food, but it was actually better for a substantial fraction of the population to eat ice cream than it was to eat rice in terms of their blood sugar. And so then out of all the factors that they measured, they looked at what actually contributed to their machine learning model. What they found was that the microbes were the main deciding factor about whether people were in the rice versus ice cream category, and so on for a whole lot of other pairs of foods that are healthy or unhealthy in opposite directions for different people. And so I'm learning this, often people have two questions. The first is there some test I can take that tells if I'm in the rice category or the ice cream category? And the real answer is no, only in the context of a research study, but it could be developed like that. But then the second question is, suppose I find out that I'm in the category of people who should eat rice, but I want to be in the category of people who should eat ice cream instead. Could I change my microbiome to get there? That might sound kind of frivolous in this example, but in the case of cancer, it's deadly serious, and I'll talk about that a bit right at the end. And we now know that the microbiome affects the impact of a lot of different drugs, including painkillers, including heart drugs, including cancer drugs, including the latest checkpoint inhibitors for cancer, like you might have heard of Keytruda, and how it can miraculously cure melanoma for about 30% of people. And we're now thinking that maybe most molecules that we ingest, whether they're drugs or food, the microbiome has a huge impact on interpreting them and seeing whether they're going to be healthy for us or not. And to scale this, we turn to citizen science. And this is Daniel McDonald, who's the scientific lead of the American Gut Project, which has now evolved into the MicroZeta initiative. And most of these projects, most citizen science projects are on the scale of a few $1,000s, but we're already on the scale of a few million and continuing to grow. And with having consistent data on a lot of people who answer the same questionnaire as well as sending in their samples, so we're using crowdsourcing and crowdfunding to scale this to whoever's interested in participating. Basically, we can ask what affects your gut microbiome strongly versus less strongly. And so the steeper the curve on this plot, the bigger impact a particular factor has on the microbiome. And so things like age and inflammatory bowel disease and what time of year you collect the sample all have big impacts, even bigger than the impact of antibiotic use. And then things like sleep duration and BMI and alcohol and exercise has smaller effects where you need hundreds of people per group to see them. But what amazed us was that a very simple thing, the number of different kinds of plants people ate, had a huge impact on the gut microbiome. And we never expected that. So eating a big mound of broccoli every night won't help you diversify your gut microbiome. If you're thinking about adopting a mostly plant-based diet, either for health reasons or for environmental reasons, it's critical to eat a wide diversity of different plants, including different vegetables, different fruits, different spices, and so on. So to build on this, we launched the Global Foodomics Project with Peter Dorrestein's lab. And essentially the goal of this is to characterize what's going in with what's going out, then integrate all the data, both at the microbial level and at the chemical level, to understand the impacts of food on health. And the sort of data we get is a molecular network. So this is a graph where the nodes are different molecules were detected by mass spectrometry. And then the edges say that the molecules are more similar in terms of the features that they have from fragmentation in a vacuum in the mass spectrometry instrument. These are sorted out by connected components in a graph layout tool called Cytoscape, developed in part here at UCSD by Trey Ediker's group. And the nodes are colored green if they came from food, brown if they came out the other end, and blue if they were found in both places. And you can see there's not a lot of blue on this plot, which just speaks to the profound transformation that happens to those molecules in food as they pass through our GI tract. And so we're taking this directly into the study of Alzheimer's on this large-scale project funded by NIH, where we're piggybacking on clinical trials of diet interventions aimed at Alzheimer's, including MIND, BEAT-AD, and US Pointer, where in all of these studies, people's diets are being modified, and then we're looking at how those modifications track with their microbiome, with their metabolomes, so all the molecules in their gut and in their blood, and also on their cognitive status. And this just gives you an idea of the large number of people and organizations around the world who are involved in this. And one of the blockbuster results from the study has been using machine learning methods to do diet readout directly from the clinical sample. So the idea is that most of the molecules on that little network diagram I showed you are unidentified. All we know about them is their molecular fingerprint. But if we take thousands of food items and run them through the mass spec, we can say, okay, we don't know what this molecule is, but we know that it came from an apple or an egg or a salmon or whatever. And so then we can take a sample of blood or a sample of stool, and we can match the molecules in it up to those known dietary sources. So this is just a principal components analysis validation that we can do this for real. And what this is doing is taking the multivariate spectral data and giving you a 2D projection of it. And you can see that on the plot, omnivores and vegetarians are different from each other. So that's basically some ground truth validity. Then when we ask why are they different, the omnivores are eating more dairy and meat, and the vegetarians are eating more legumes, fruit, and so on. So this just shows that you get what you expect out of this analysis from the molecular data directly. You don't have to ask them, what do they eat specifically, which people are really bad at writing down. So what we can then do is we can take those spectra and we can correlate them with things like memory. And so this heat map is showing you positive versus negative associations of each type of food with the 4 subscores on a standard measure of learning. And so what we see is basically that the foods that are bad are things like sugarcane and soda. And then the things that are best for memory are things like fish, strawberries, and mushroom. So you might wonder, well, so what? Couldn't I have pulled any food out of a hat and it would seem equally convincing? How do I know that this is actually real? But when we look at the literature, what we find is a giant literature on how soda is bad for memory and bad for Alzheimer's risk. Same thing for sugar. Giant literature on problems, on cognitive problems and links to Alzheimer's disease with sugar. Then at the other end of the spectrum, what we see is a huge literature on fish being good for your brain, improving memory, and preventing decline in the elderly. We also see some evidence for strawberries and some evidence for mushrooms in a smaller scale, but controlled studies. So what's awesome about this is that we can take literally thousands of person years of individual studies and turn them into an afternoon using the mass spec to be able to read out directly what correlates with health and what doesn't. And we're really excited about the potential of this to be used not just in the context of cognitive disorders, but all kinds of other health conditions like diabetes, like major depressive disorder, like sequelae of HIV, and all kinds of other things that we're studying here at UC San Diego. But even with machine learning, there's still a big gap when you have specialists in machine learning trying to communicate with physicians who are still very skeptical of this stuff. where they have questions like, what does classifier accuracy mean? They never taught me that in med school. They have questions like, are these features causal, or can you just, or are they just correlations? Like suppose having a healthy brain makes you want to eat salmon rather than salmon makes your brain healthier, and so on. Or, correspondingly, maybe an unhealthy brain makes you want to chow down on all that sugar and soda. And then trying to understand which species or molecules are really making the difference. When you pull out the features that contribute to accurate classifiers, there's a lot of masking. So if you have correlated features, you're going to pick one at random to report rather than picking all the correlated ones. And that, again, is very annoying to physicians if you pick one that they don't know automatically from the algorithm rather than one that they're very familiar with. And then finally, how to explain this stuff to patients. If you're trying to get them to explain all the details of a random forest classifier, how you did the cross-validation, how you did test versus training sets, all that kind of stuff, that is exactly the sort of thing they do not want to have to describe to a patient in like the 8 minutes they have together. So again, we still need something easier than that. So the third transition that's happening at the moment is harnessing AI to solve a lot of these problems. And so we've been using AI for quite a while. Back in the Human Microbiome Project days, so 2007 to 2012, we were still using machine learning. And already at that point, it had been evident that we were going to need to give up on the human brain and move to automated techniques for a lot of this stuff. And back in 2017, we launched an agreement between UC San Diego and IBM to form the Artificial Intelligence for Healthy Living Center, which I've co-directed since 2017. And this is part of IBM's AI Horizons network, so a network of similar centers around the country. And some microbiome patterns are really straightforward, and we can decipher them even with our puny human brains. So if I show you this graph, you can see immediately ages on the x-axis, the distance between each sample and the average of adults in the population is on the y-axis. And you can see right away that the samples start off very different in the younger kids, and then they converge by age 3, and then basically flatten out. So you don't get any more similar to the adults all the way through 18, or indeed, when we extend this graph, all the way through age 80. So you can just see by reading off the graph that 95% of the gut microbiome change happens in 2.5 years, and you don't need any AI for that. But what does this mean? Does this mean that your microbiome is doomed by age 3 and the choices were mostly made for you by your parents? That wouldn't be very good, right? So to figure it out in adults, we turn to the American Gut Project data set, where we've got over 25,000 samples in the training set. And the microbiome changes with age in adults are much more subtle. So these are the scatter plots of age versus a few measures of diversity. And you probably can't see any pattern in that. And if you run a regression line through it, you can get regressions that are statistically significant, but they hardly explain any of the data. And you definitely should not believe the parameters of those curves. So this is where we have to give up on the human brain and turn to AI instead. So our first attempt at this was with machine learning. So what we did is we did random forest regression to ask, if I have a biospecimen from different parts of the body, can I tease out biomarkers with age from those different parts of the body? So when we do this from poop, we can get your age to within 12 years from a sample of your stool. That's not too bad. We do it from a sample of your saliva, we can get it to within 5 years. And most remarkably, if we do it from a swab of your skin, we can get that to within just 4 years, which is pretty amazing, right? I can swab your forehead or the palm of your hand and tell your age to within 4 years based on the DNA of the microbes there. So then the question is, how does your microbiome age compare to normal age? And so the idea is that as you have this process of aging, we've shown now that you have this clock-like behavior between chronological age and microbiome age on average. But what about the residuals from that regression? What about the individual variation from it? And so the idea is that maybe if you have a young microbiome age, but an old chronological age, maybe that means you're extra healthy. Whereas in contrast, if you have an old microbiome age for your chronological age, maybe that means you're extra unhealthy. And we're doing that with the FinRisk data set at the moment. So people who pooped in a cup for science back in 2002 in Finland, and we have all their health records all the way to the present. So we can tell from their microbiome all of the bad things that happened to them over the intervening 20 years. But Kaylin Cantrell, who's one of my CSE grad students, has found that transformer models radically improve this age prediction. So this is just a reminder of what I showed you before. And with the transformer models, what we see is the error from the gut microbiome is just 4.5 years. It's just 2.7 from the saliva, and it's less than 2 from the skin, which is truly remarkable improvement. So we're now applying this sort of thing across a whole lot of different things, including cancer, development, neurological disease, and so forth. Let's see. This is running a bit longer than I thought it would, so I'll just summarize this part really briefly. So Larry Smarr is one of our collaborators who we've worked with for a long time, and nothing I'm going to tell you about violates HIPAA because he's very aggressive at publishing articles about this in the popular press like The Atlantic. And so shortly after I came to UCSD, I was talking to Larry about the Human Microbiome Project data, like I showed you, only this was on the 64 million pixel display wall over at Cal IT2, which is about a 10-minute walk across campus from the building in the med school where I'm sitting right now. And Larry wanted to get himself on that plot. And so shortly after that, this box showed up in my lab. I told you he was a pretty interesting person. That was just the first of many. But what we found is that over the course of 8 years, Larry became 5 different people in terms of his microbiome, as he did things like got colonoscopies, had surgery, and then went on a vegan and gluten-free diet and on a time-restricted feeding diet. And what we found was that, fascinatingly, the time-restricted feeding plus a smoothie with 60 kinds of plants on it. You might have seen this on the Hack Your Health documentary from Netflix recently. That smoothie had a bigger impact. That's Larry number 5 on his microbiome than being vegan and gluten-free for months did. And that's pretty amazing. So then the question is, can we take existing data and produce high-resolution predictive human digital twin models? And at the moment, our models are pretty crude because we're mostly using short-read data and not very much of it. So we get like this little pixelated troll of Larry. Then the question is, could we turn it into a beautifully synthesized image with generative AI with much more predictive power that contextualizes him in his environment and so on? And we're working on bringing together a combination of technologies to do that right now. One really important part of this is Nutrition for Precision Health program. So this is a $170 million investment from the NIH Common Fund, where basically the idea is to develop new algorithms that predict individual responses to foods and dietary patterns, like I showed you from the Israeli group, but on a dramatically larger scale. And it's got 3 modules. The first is characterizing 10,000 people cross-sectionally, looking at their response to a standard diet. And then module 2 and module 3, crossover studies where you vary the diet, look at how people respond. One's a behavioral intervention at home where you tell people what to eat. The other is an inpatient study where you give them exactly what they have to eat and measure it and watch them the whole time to make sure they do it. But the real problem with all this is even when we have that data, we need a better user interface for our microbiomes, because the last thing you want to look at is complicated plots or derived statistics from machine learning, or worst of all, a list of 1,000 Greek and Latin names of species that you've never heard of that are somehow playing an important role in your gut. And what our dream is to take this into settings where people can use them in daily life. So the dream with this is that you could scan the barcode of a yogurt when you're confronted with 1,000 different brands in the supermarket, and then that you could interact with it with natural language. So I'm sure you've all used ChatGPT by now. We're calling this system Crap GPT, where the idea is that you could stand there in the supermarket and ask your phone things like, should I eat this product to improve my health? Or as a stretch goal, can you highlight with augmented reality the products that are on the shelf in front of me that I should eat instead of this product? And that might sound like absurd science fiction, but the problem of detecting which out of a relatively finite list of products is a particular product you're pointing at on the shelf is a much, much, much easier computer vision task than a lot of others that are being solved at UC San Diego by our colleagues right now, especially with things like self-driving cars, self-navigating drones, and so on, that can see anything in the natural environment. So the constrained space of a supermarket shelf is a much easier target to start off with. And the UPC, the barcode on them, is linked to all kinds of nutritional information that we can use to inform these models. So that's the kind of stuff, that's the kind of crazy stuff we're up to at the Center for Microbiome Innovation right now that I direct at UC San Diego, where we have over 140 faculty at UC San Diego who are affiliated with the center in one way or another, working on all kinds of things like this. Now, I promised you the non-frivolous version of that yogurt versus ice cream thing. And so Jen Wago at MD Anderson Cancer Center is using microbiome-based classification right now to figure out whether patients are going to benefit from Keytruda, one of the checkpoint inhibitor therapies for melanoma, and literally whether they'll live or die. And she took this a step further by asking, suppose I run this test and I find that your microbiome is bad for Keytruda. Could I modify your microbiome so that Keytruda is going to work and so that you're going to survive your melanoma? And so in this article in Science a couple of years ago, she studied the influence of dietary fiber and probiotics in changing the microbiome so that you can have a better immunotherapy response. And basically what she found was that fiber was very effective. But with probiotics, the patients taking probiotics actually died faster, although it wasn't statistically significant, but they had a worse survival curve. And then what she found is that you could take the fecal sample and put that from humans into mice, and then also give them probiotics or withhold those probiotics, and then inject them with melanoma cells from the patient. And so what she found, and that's the graph in panel C, what she found was that the tumors got bigger and the mice died faster if they received probiotics than if those probiotics were withheld. And this is a really important, sobering message, because just because people think something's healthy or just because it's fine in a general population, that does not mean that you can apply it in clinical populations. And we have to be much, much, much more careful about doing tests in those clinical populations rather than letting doctors and patients go with common sense recommendations, which may not work at all when you're talking about people who are immunocompromised or otherwise sick. So where we think the future of this is the technology developed by Amir Zaranpar, another of my colleagues here at UC San Diego, where the idea is to take microbes that already colonize a person and then grow them in the lab and then modify them and put them back into that individual. So it's sort of like CAR T cell therapy, only with the bacteria instead of with T cells. And we think this has tremendous potential for modifying the metabolism of particular substances in the patient while leaving the rest of the microbiome alone and not having the bad influence that you have with probiotics. So finally, we think we can deliver something useful to physicians out of all this through some combination of translation and upscaling of different kinds of microbiome data. We hope that we'll be able to give explainable and accurate and personalized predictions using AI. We think we'll be able to design new microbiomes, diets, and therapies, including the identification of active ingredients in diet, like how do you turn that number of plants result into what particular plants should you eat to have a big impact. And then finally, we think the natural language interaction is going to radically transform people's ability to interact with all this microbiome data. And all of this is happening very fast. So just to give you an idea of the history of AI image generation, in 2016, the idea that you could do text-to-image synthesis led to this bird generator that could make 64 by 64 pixel pictures of birds with very restrictive settings just from the text. Whereas now you can basically type any text you want into Midjourney or Stable Diffusion or DALL-E 2, get these beautiful photorealistic images in whatever style you like. And that's just 8 years of progress. And all these hard problems are falling to generative AI at the moment, including protein folding, including weather forecasting, and including physician-patient communication, which is already being done here throughout UC San Diego Health. So all these hard problems are already being solved by very rapidly by improvements in AI. And we think these problems in the microbiome will also be susceptible to similar approaches. One quote that I really like is from the science fiction author William Gibson. He likes to say that the future is already here. It's just not very evenly distributed. And a lot of the things that I've said that sound particularly futuristic are already being applied routinely in some lab or another at UC San Diego. So a lot of what we're doing at the moment is bringing them together so that we can do something truly revolutionary and groundbreaking. Anyway, so with that, I have to acknowledge a huge number of people who've worked on this stuff with me, including over 1,000 collaborators in different labs, including the numerous people in my lab here at UC San Diego, and a lot of sources of funding that we're very grateful to, including the tens of thousands of members of the public who've contributed to our citizen science efforts. And with that, I know we're a bit over time from the half hour that I promised Nima, but thanks for your attention and for sticking with us, and I'd be delighted to answer any questions you have. Thank you.
Awesome. Excellent talk. Thank you so much. That was quite a journey. We went over diabetes, health, ice cream, rice, cancer, poop, poop from your boss, all sorts of things.
Yeah, imagine if you could cure diabetes with ice cream rather than causing it. That'd be pretty cool.
Oh yeah, that would be game changing. Any questions from the audience? Please feel free.
Yeah, usually people have a lot of questions about this stuff, so don't be shy.
Yeah. So, hi, my name's Max. I'm a rising sophomore now, majoring in computer science with a specialization in bioinformatics. And really, I've kind of been, working through a lot of your tools. Actually, my first exposure to coding was working with Nima on the Cheetah platform with plugins for COVID. So it's been amazing being able to use all your software tools. And now that I kind of have you here in front of me, I kind of wanted to ask you how, like in the next, 3 years, because that's when I'm here at UCSC, how do you see like that developing? Like, is it really bounded by compute or is it more by bio? Like, yeah, like what do you see in the coming years?
Yeah, so there's three boundaries. One is data generation, which we're working on very actively, especially scaling up long read technologies, which we're really excited about. Second is algorithm design. So especially we've got a group of people who you'd be welcome to join who are working on things like transformer models, attention models, upscaling from 16S to shotgun metagenomic data, transfer learning to understand what you can learn about the metabolism layer from the microbiome layer and vice versa. Can you bridge from a blood sample to a stool sample, those kinds of things. Hardware access isn't too much of a problem at UCSD. We have access to quite a lot of resources, both on the GPU and on the FPGA side through SDSC. And then I have my own cluster that's a few thousand cores, mostly CPU for more traditional microbiome workloads. There's also a critical need to identify training data that's published in the literature and get that into an AI-ready form. So that's something that that's something that we're starting to do a big push on our NCI project. That's about integrating cancer data sets and understanding the role of the microbiome in cancer broadly, which is a collaboration with Diane Simeo, the new director of Moore's Cancer Center, and with Greg Capareso, who I mentioned during the talk, who was the original lead developer of CHIME.